Hello, it's your boy, the one and only Third of the Department of Biology. So I'm just going to read some announcement from our BCAA. So in the meeting of the Youth System Academic Affairs Committee yesterday, it was agreed that there will be no suspension of classes this week. Well, but in the context of surging COVID cases, the ECQ in NCR areas, and GCQ in Biocity and WCU, all faculty members are enjoined to be more flexible in the understanding of students, their concern to waive attendance check for remote classes and to meet synchronous meeting at this time. So the official memo from EPAA will follow very soon. But for now, instead of doing our synchronous classes, I will record asynchronously for you to follow. So without further ado, welcome to Lecture 4, Identification and Classification of Microorganisms. So before we start, let's just have some simple learning objectives for Lecture 4. So the first one is to discuss the purpose of identification and classification in the world of microbiology. So remember your good old days of biosystematics. So at the end of class, you should answer the question, why bother identify these microorganisms? What's the purpose of identifying this microorganism? Why are we spending a lot of time, effort, and energy and money to identify and know the names? Identify these organisms, this microorganism around us. And the next objective would be to describe these different procedures that we use as microbiologists to identify these organisms, these microorganisms. So that's the two main learning objectives for this class. Now moving on, okay, so you can see here. So again, the question is, why bother? Why bother classify and identify microorganisms? Well, we as biologists and future microbiologists classify organisms for several reasons. The first one is to bring sense to the world around us, to bring order and organization. Again, we humans are nitpicky. We have this urge within us to put things into certain boxes that we call clades or taxa. We have this urge to put these organisms, these microorganisms, these species into different hierarchy and levels. As you have recalled from your biosystematics, we have your family to genus to species. We like to box these organisms into those categories, into those hierarchies. We humans are like that. And the next one is to make predictions on the structures and functions of similar organisms, similar or related organisms. So because more likely if those organisms belong to a certain taxa, they will exhibit similar characteristics. And that's good for us, no? That's good for us who would like to explore them. I mean, exploit them. Wink, wink. Yes, because we humans are utilitarian in nature. But aside from that, the next one is to uncover and understand the potential evolutionary connection across different taxa, no? Because nothing in biology makes sense except in the light of evolution. That's Theodosius Dobzhansky, 1973, no? Kung maalala nyo pa si biosystematics nyo. Anyway, so yeah, it's an effort of us humans to find out where we came from. No? Find out who and what our ancestors are. How these different life forms on Earth are related. It's an existential and a scientific journey at the same time. So again, so you have recalled taxonomy involves declassification and nomenclature plus the identification of your microorganism. But the difference of taxonomy and systematics, also in microbial systematics, is that we're also looking at the tree of life, no? The phylogeny of things. Okay, so let me just bring back the memory of Carl Woese and the three domains of life. So in the three domains of life, we have bacteria, we have archaea, and eukarya. So this is the major scheme of classification that we're currently, until now, we're using. No? So we have the universal common ancestor that branch out into different domains, into the different domains of life. And you should note that these mitochondrion and this chloroplast here made a jump, no, through the eukaryotic lineage by a process of what we call that eukaryogenesis or endosymbiosis, right? Where the bigger cell ate the smaller cell, and the larger cell didn't digest the smaller cell, and eventually they live symbiotically together. No? But as you can see here too, no? The branch of your archaeal lineage is more related, no, to your eukaryotic lineage. As you can see here, these two domains are more related to each other, no, than your bacterial lineage here, that branch off early on in the tree of life, right? And I'd like you to look at that more carefully. And so this is the older and more simpler version of the tree of life. Let me just show you the current view of the tree of life that we're using here in microbiology. Well, this is not that current because I think this was released back in 2016. So it's about 5 years ago. But the thing about this tree, this current view of the tree of life, is that it encompasses the total diversity represented by the sequenced genomes of your archaeal, your bacterial, and your eukaryotic representatives. This is actually based on what we call the 16S and your 18S rDNA sequences. Okay? So this is based on gene sequences. So as you can see here, if you look at the tree, we have at least 92 bacterial phyla. And I think about 26 archaeal phyla right here. And 5 eukaryotic supergroups right here. Okay? And if you look closer to this tree, no, it's based on the complete ribosomal protein tree, right? Because it's based on your 18S and 16S rDNA sequences. And by looking at this tree, you can see that in terms of rDNA sequences, ribosomal DNA sequences, your archaeal lineage are more closely related to your eukaryotic lineage than, of course, your bacterial lineage way up here. Okay? This is what I call the anahaw tree. It looks like an anahaw, right? It's fanning out and weird looking. And if you look closer too, no, we have this portion right here. Loki, there's Loki, right? Loki, the god of mischief. The Bifrost. Yeah, the Marvel Cinematic Universe Loki. Yeah, it's from that character, from that mythology of your Norse mythology. It's German, right? Norse mythology. But yeah, so we're closely related to our Archean. Those archaea. No, the archaeans are closer to us, no? Eukaryotes, and specifically these Asgardian, what we call the Asgardian lineage, no, the Asgardian clade or Asgardian taxa. Asgardian taxon, no? Okay, so let me just share, because I have read across different papers. in 2019, several Japanese researchers have isolated an organism, an archaeon, at the prokaryote-eukaryote interface, what we call the prokaryote-eukaryote interface. So the interface between your prokaryotic organism and your eukaryotic organism. And they have isolated this archaeal representative under the Asgardian taxa. And the amazing thing about this study is they are challenging the current endosymbiotic theory that Lynn Margulis proposed back then. No? They are actually challenging that notion of endosymbiosis. For them, it seems like it's impossible for an organism, if you put your context, if you put the context in the primitive world where energy is super small, the amount of energy for a cell, for example, is incredibly small for it to perform that phagocytosis mechanism. So they think that it's impossible for our ancestor eukaryotes, no, to engulf that bacteria, that mitochondria, that chloroplast, because it's impossible for it to do that since it lacks the energy to do phagocytosis in the first place. So they are proposing what we call the E3 model. Okay? So the E3 model is what we call the entangle, engulf, enslave model. The E3 model. I can send you the link later for the paper if you want to read more on the matter, or if you're a Marvel fan. But yep, the paper have proposed that eukaryotes have arisen from an archaeal lineage. More specifically, the archaeal lineage, the taxa known as the Asgard archaea taxa. No? This Asgardian taxa, this Asgardian archaea. Okay? So this Asgardian archaeal lineage includes not just your Lokiarchaeota, not just this Lokiarchaeota, it also includes Heimdall archaeota. It also includes Odin archaeota. And of course, your Thor archaeota. It's Asgardian. Those are Asgardian names from your Marvel Cinematic Universe and from the Norse mythology. So those Asgardian archaeon, those Asgard archaeon, they propose are a close relative of the eukaryotes. Let me just show you this figure from the paper of those Japanese guy. So here we have your Loki archaeota, Thor archaeota, or Odin archaeota, Hel archaeota, and the Heimdall archaeota. All of those Asgard archaea are a close relative of us eukaryotes. And from here, they have proposed what we call the E3 model for eukaryogenesis, the entangle, engulf, and enslave model that challenges the current notion, the current hypothesis for eukaryogenesis, the endosymbiosis of Lynn Margulis, where our eukaryotic ancestor, have phagocytosized, have eaten this small cell, right? So they're challenging that notion of Lynn Margulis. What they're proposing is that because what they have seen from the Asgard Archaean that they have isolated is that this Asgardian archaea produces what we call cellular protrusions like this, somewhat a network-shaped protrusions of the cells. So this is the characteristic of your Asgard archaea that they have isolated. So what they're proposing is that our pre-least eukaryotic common ancestor, our ancestor, was an Asgardian archaea that may have produced such protrusions, and those protrusions of that archaeon may have allowed for the growth of your a symbiotic growth, no? A syntropically growing near each other of another species of bacteria. And essentially, they will live in close proximity to each other, and essentially that bacterium, will be trapped inside this net-shaped protrusion of your Asgard archaea, and eventually they will fuse together, and the bacterium would be trapped inside. So it will be entangled by the net protrusion of that Asgardian archaea. And then eventually, the protrusions may have fused inadvertently and will surround the partner altogether. So this mode of eukaryogenesis is phagocytosis independent engulfment, because it will trap it first. Then after some time of codependently living, syntropically living and growing together, they just fused. And essentially, the bacterium gets to be enslaved inside. So that's the model that they're proposing. But anyway, that's going far ahead. I'm just a Marvel fan. That's why I would like you to read on this, because we scientists, we taxonomists can do that thing that we call tadhana, where we name those organisms, the taxa that we're playing with. Yeah. So yeah, you can think of more clever names if you want to go into taxonomy. But yeah, share lang. Moving on with Polybasic Taxonomy by Rita Colwell. So Rita Colwell proposed the use of Polybasic Taxonomy, where it contains at least 3 stages in describing and in the identification and classification of microorganisms. Basically, it includes 3 processes. The first process is to analyze the phenotypic characters of that organism. Basically, a lot of phenotypic characters, the cell shape, cell size, arrangement, number of flagella, so on and so forth, type of cell wall, gram-positive, gram-negative, acid-fast, positive, acid-fast, negative, endospore, spore formers, non-spore formers, and so on and so forth. The next step in the identification and in the taxonomy of microorganism would be the characterization of the genomes of that organism. It can be in the form of metabolic genes of that organism, or it can be in the form of your 16S RNA, the 18S RNA, those RNA genes, rRNA genes, the ITS, internal transcribed spacer for fungi, usually we're using that. And we're also using barcoding analysis, no? For organisms that don't have a barcode yet, don't have a what do we call this, a tag in the database of NCBI. Well, moving on with that is the use of your phylogenetic analysis. Of course, in phylogenetic analysis, we refer to the position of the organism in the evolutionary perspective. So that's Rita Colwell's Polybasic Taxonomy, which has 3 stages. So we'll discuss that in detail, starting with phenotypic analysis. The first examples of phenotypic analysis are, of course, your physical characteristics. This includes cellular morphology, and there's a problem with cellular morphology, if you know what I mean. Because, for example, if you use Linnaeus scheme for cell morphology of your bacteria, he only pertains to at least 2 schemes. No? For his classification, he uses Bacillus and Coccus. So you can already see the problem here, right? If you're just using this physical character, if you're using the classification scheme of Linnaeus back then, he's just using Bacillus, Coccus to identify and characterize organism. And that's problematic, because that would lead to a problematic group and a paraphyletic clustering. A very non-functional clustering, actually, because phylogenetically speaking and evolutionarily speaking, those Bacillus and those Coccus organisms may or may not be related to each other, actually. they may just look the same, but they're not related to each other. I mean, okay. So that's why I actually lecture ahead on the physical requirement for growth, so that you know the physical characteristics of your organism and their physical requirement of the organism, and that's among the physical characters that we're looking at. If they are halo tolerant, if they are, well, they are what we call, they can survive. No? They are aerobes, they are facultative anaerobes, things like that. No, those are the physical characteristics that we are referring to when we use phenotypic analysis. Okay, so aside from your physical characters, that is quite problematic. We also look, yes, these are the examples of some phenotypic characters that we're using in microbiology: morphology, motility, metabolism, physiology. And if you look at this portion, no, we are also looking at the cytoplasmic membrane characteristics of different microorganisms to describe them. Usually, we ask the question, what fatty acids are in their cytoplasmic membrane, what amount, how much those fatty acids are present. Different fatty acids are composing the outer covering of your organism, of your microorganism, and of course the other macromolecules in that cytoplasmic membrane. So those are some phenotypic characters of taxonomic value. So yeah, as I was mentioning earlier too, the presence of flagella is also important. The gram reaction that we already discussed earlier, the colony morphology that you will later on see, the presence of spores, the inclusion bodies, like your polyphosphate granules, your magnetosome, your vesicles. Those are your inclusion bodies that are, again, highly refractile. So you must use endospore staining to differentiate them. Anyway, so yeah. And on terms of metabolism, we can zoom in more on that in terms of your biochemical tests. So for biochemical tests, again, still under your phenotypic characters of your microorganism, we have what we call substrate. And your organism will have certain enzymes, of course, that will act on that substrate to produce a reaction on our test. No? And this reaction could be in the form of waste product, could produce waste products, could like your hydrogen sulfide. Oh, sorry. H2S, hydrogen sulfide, presence of breakdown materials, cellulasic materials, the things like that. So yeah, for the biochemical test, we use different substrate and we pin on the differences of the metabolic activity or the ability of your different microorganism to degrade or to utilize those substrate and produce a reaction product. So basically, in biochemical tests, we're looking at the waste product, the reaction, the change in color, the formation of gas bubbles, the formation of lactic acid, for example, the production of certain chemicals. We're looking at certain changes in the reaction tube to see if that certain microorganism has a positive reaction or a negative reaction for certain substrate, if they can utilize it or not. Okay? So that's your biochemical tests. Examples of your biochemical tests are here. No, we have your negative carbohydrate fermentation, positive carbohydrate fermentation, and fermentation and gas production. So in this case, what we're doing is this is the substrate, right? Initially, the color of this substrate is pinkish, but after inoculation with the organism and after incubation, if that organism can ferment or utilize that carbohydrate, it can change color or produce reaction bubbles, no? Gas bubbles. Okay ba? So it can produce certain black tinge, silver lining tinge on your substrate. Things like that. We're looking for things like that for us to conclude if it has a positive or a negative reaction, okay, to that substrate, for example. In this case, your carbohydrate fermentation. Okay, but a more streamlined approach, and but we're not using it here in our class. What we're gonna do in our class and in your laboratory portion is just this portion where we're using test tubes, and it's a lot difficult to do it actually, because you need to prepare the materials, you need to prepare the carbohydrates, the proteins that the organism will degrade, and you will incubate that microorganism inside that test tube, and so on and so forth. So it takes a lot of effort to do that. But a more streamlined approach for biochemical tests would be the use of Analytical Profile Index, or your API. The Analytical Profile Index simply is a miniaturized biochemical test, no? So it's a miniaturized panel with so different panels contain different substrates, and what we're going to do is we're going to inoculate that microorganism in each panel. Okay, so after inoculation, we're going to incubate them. After some 24-hour period of incubation under 37 degrees Celsius and so on and so forth, we're gonna do a pointing system. In this pointing system, as you can see, each point will correspond to certain number. So if it has a positive reaction in tube number 1, it will get a 1 point. If it has a positive reaction in tube number 2, it will get 4 points. And essentially, we're going to see the reaction if it has positive or negative in that section. And we're going to assign a point to that. And we're going to sum those points. And we're going to add those points. And we're going to get this 7-digit code. So that's your Analytical Profile Index, or your API. When you have this number, you can just put it in the database, and it will automatically tells you the identity of your organism. So what are these are the classic substrates, and the reaction that is being tested on those different miniaturized panel right there. And as you can see, we're looking for the positive and negative results right here. If it's colorless, if there are color changes, the production of bubble, production of particulates, it's just right here. If you are in my biosystematics class, you may have encountered the API index, Analytical Profile Index. But for our case, back then, I actually didn't let them use the system, the API system. Because if you can see here, if I log into that database, if I click that, as you can see here in the database, you need to have an account. But to have an account here, you must have a registration code from the miniaturized panel itself. If you need to order the Biomerox API miniaturized panels for you to have the code, so you can log in and use their database. But if you're rich, you can just do that. You need to buy the panel, the miniaturized panel. You perform the test, you incubate the bacteria, you input the 7-digit code, and voila, you have the identity of the organism. But if you are poor like us, you can just go to this website, the ABIS online database. This database is free, and you can perform the same biochemical test. So it's just a database. I'm gonna put my pseudonym here, my game name. There you go. I'm just gonna log in, and as you can see here, we have different tests, biochemical identification tests. So you just need to narrow your microorganism of interest. You can have different family here, Enterobacteriaceae, Bacillus, Bifidobacterium, Clostridium, so on and so forth. Let's just say it's an Enterobacteriaceae. And what we did back then in my biosystematic class here, it's just a simple test. You also need to perform the different biochemical tests, the presence of the activity for oxidase, different activity for different carbon sources, degradation fermentation process of different carbon sources. We have utilization of different amino acids too. You need to perform the biochemical test, but the easier thing here is that it's free and you just need to take the characteristic of your microorganism that you have performed the different biochemical tests. Again, you need to prepare different substrate here. You need to perform every test on your own. You need to prepare every material and substrate, but you don't need to pay that much. You can use the equipments here in our laboratory. You have different reagents in Manang A. You can use that to perform these different biochemical tests. You don't need to perform everything. You need to perform most of them to enable you to limit and narrow the tests and to pinpoint what organism you're using. You have the identity of the organism that you're dealing with. So yeah, this is just the tick system. You need to tick if it has a positive or negative reaction for that certain biochemical test. So that's that, no? You can just go to my account and just find the activity tree, API biochemical tests, and the ABIS online database. I had to scour the internet for this database, but yep, that's the database that I'm using if you're into biochemical tests. Okay? So let me just close this and we're back here. So moving on with our lecture. Another example of a biochemical test is what we call the Biolog microbial ID system. For the Biolog microbial ID system, this is a 96-well microbial ID system. It contains different carbon sources. Okay, and it will check if your organism has a metabolically active reaction for that substrate. So it contains different substrates here. So it's a 96-well plate. You will inoculate your organism in that plate. So what's happening here is you will just check for the change in coloration. Okay? If it is positive, it will get the color of purple. If it's negative, it will be colorless because your Biolog microbial ID system relies on Biolog redox chemistry, wherein it has here, it has a redox dye here, where a positive reaction would change the color of that redox dye. So from colorless, it will turn into a purple color. Okay? Colorless. So these are the positive reaction, these are the negative reactions right here. Okay? So that's your Biolog redox chemistry. Unlike your API, this is much more streamlined and it has much more reagents and substrate here. So you can narrow the identity of your organism much more. Okay? So that's your Biolog microbial ID system. And then again, for your phenotypic classification, for your phenotypic analysis, we also have your serological test. For serological tests, here we take advantage of what we call antigen-antibody reaction. Because as we all know, antibodies are specific to certain antigens. These two have a very specific, kind of like a lock and key interaction with each other. So what's going to happen here is for a certain strain, of a bacteria, for example, so for a certain strain of bacteria. So we have here a rabbit. Say we have here a rabbit. We have here a rabbit. Okay, so this rabbit will inject the attenuated typhoid, for example. Attenuated typhoid. Inside that rabbit will inject the weakened typhoid causing the organism, okay? And after injecting that attenuated typhoid there, we're going to extract the anti-serum of that rabbit from the bloodstream of the rabbit, for example. And what we're going to do with this anti-serum is we're going to concentrate the antibody that is specific for the reaction for the typhoid. After isolating the antibody for typhoid, we're going to concentrate this. We're going to concentrate this and we're going to put it in the bottle. And then we can do now the agglutination test. So what's agglutination test? Essentially, it's the formation of clumping. It's clumping, okay? Formation of clumps, agglutination test. You check for the formation of clumps because as you remember, your antibody will respond to that specific antigen. For example, the antigen of your typhoid, okay, will respond to that antibody, that concentrated anti-sera, okay? So what's gonna happen is that it will form white clumps or clumping reactions right here because the antibody will surround that antigen, that bacteria. Huh? It will surround that antigen. So if that antibody for typhoid reacts to this, or so for example, E. coli 2 or Pseudomonas or any microbes you're using that you have the antibody for, you just need to put in a drop of that anti-sera and you watch for agglutination. If there is no agglutination, this is negative, so it's not that organism. If it has clumping, presence of clumping, it's positive. So meaning it is that organism. So if you have an anti-sera for typhoid, if you have anti-sera for E. coli, if it is positive, if it has clumping, it is that organism. You can confirm that it is that microorganism, okay? You just look for the presence of clumps. Moving on, we also use what we call page typing. Here naman, just like in your antibody and in your serological test, your anti-sera, in page typing, we use the specific relationship. We take advantage of that specific relationship between your bacteria and the bacteriopage. So again, bacteriopages are those viruses that specifically attack certain species of bacteria. These pages are species specific. So we can use them to characterize the host, the strain, the isolates that we're using. So what we're going to do here is we're looking at the page reaction. Okay, we're looking at the page reaction. The reaction of this page to the bacteria. So what's going to happen here is we're going to inoculate bacteria on our agar plate. So we call this the bacterial lawn. And we're going to drop this, what we call the pages, no? The bacteriopage in that bacterial lawn. And we're going to look at what we call plaques. These plaques are technically are zone of lysis. If it has this zone of lysis, if it has these plaques, it means it's positive. It means the page that you dropped right here, the bacteria page that you dropped here, was able to lyse or kill the bacteria here. So you can confirm it if it's that species or not. based on the presence of plaques. Okay? So why is that your bacteriophage or your pages have a specific interaction with your bacterial host? Because as you remember, these tail fibers, the tail fibers of your bacteriophage can recognize your bacteria, the surface of your bacteria, the membrane of your bacteria. So only the specific bacteriophage, okay, for example, the bacteriophage that infect E. coli can only infect E. coli and not Pseudomonas aeruginosa. Because these tail fibers here are species specific. It can only recognize the proteins and the sugar molecules in that the surface of that bacteria. Okay? So, if I have a bacteriophage, if I have a bacteriophage solution, so this is the dropper. If I have a bacteriophage solution for E. coli, I drop it here and it doesn't form any plaques. That means that organism is not an E. coli. If I have a dropper again and a bacteriophage that is specific for Pseudomonas aeruginosa, and I drop it in that lawn again and it forms plaque, I can confirm that that bacteria that I grown here is a Pseudomonas aeruginosa. Okay? So that's simple. Page typing is. Moving on, for the last part of your phenotypic characters, phenotypic analysis for identification of microorganism, we have what we call fatty acid profiles. Specifically, we're using what we call here as your FAME analysis. What we mean by FAME, fatty acid methyl esters. So this fatty acid methyl ester analysis, FAME analysis, takes a look at the differences between the composition of the cell membrane of the fatty acid in the cell membrane of your microorganism. So different bacteria have different amount, different profile of fatty acid. Some bacteria will have saturated tetradecanoic fatty acids in their membranes. Some have more unsaturated fatty acids. Some have cyclopropane. Some have branch fatty acids. Some have, and so on and so forth. Okay? There are different classes or fatty acid types in the walls, in the cell membrane of your bacteria. So FAME analysis takes advantage of that and uses that profile, the profile of these fatty acid profiles, in identifying, fatty acid profile in identification of that microorganism. So how do we create that fatty acid profile? Essentially, again, we have your bacterial culture. We extract the fatty acid from the cell membrane of that bacteria. We convert it and derivatize it to form methyl esters. Again, fatty acid, if you have remember, are carboxylic acids, right? With RC OH, a carboxylic group. Fatty acids have carboxylic group, of the ends of your fatty acids, right? Have a carboxylic group. So what we're going to do here is we're going to replace it with a methyl ester. So instead of a RC double band OOH, we're going to make it an RC double band OOR prime. Okay? So again, this is a carboxylic group. This is an ester. but the R here is a CH3, is a methyl. So we convert the carboxylic group, those fatty acid, into a derivative methyl ester. And of course, this derivative form, this methyl ester form, is volatile. Thus, we use chromatography to create this profile right here, these peaks right here. And these peaks, of course, correspond to certain groups of certain classes of your fatty acid methyl esters. And this profile is what we're going to use to compare it to the database that we have. So we compare it to other profiles of other organisms. If it matches the profile of other organisms, it has the same peaks, we can confirm the identity of the microorganism. We compare the pattern based on the pattern of the fatty acid methyl esters, the profile of the fatty acid methyl esters. Okay? So another example of this is cell wall analysis, in which aside from your fatty acid, the fatty acid in cell membrane, we also look at the cell wall based on other chemotaxonomic marker that are present in the cell wall of your microorganism. So things like that, okay? So that's your FAME, or fatty acid methyl ester analysis. Now we move to stage 2, which is on the use of genomic analysis. The first example that we're using here is DNA-DNA hybridization. So for DNA-DNA hybridization, what's happening here is we hybridize, okay? We hybridize or pair different DNA of microorganism. And what it does is it enables us to have a rough index of similarity of the DNA composition that is inside the whole genome of that organism. So the thing about DNA-DNA hybridization, the use of DNA-DNA hybridization, is that it's more powerful than just using the 16S rDNA, because for your DNA-DNA hybridization, you're using the whole genome of that bacteria and comparing it to the whole genome again of another bacteria. So what's going to happen here is we're going to extract the DNA of organism 1 and organism 2. We're going to extract their DNA. We're going to radioactively label with radioactive phosphorus only organism 1. Then we will, okay, after labeling and shearing the DNA into smaller bits, we will perform heat denaturation and convert it into single strands. Now, we will perform 2 hybridization experiments. The first one is the what we call 1 times 1, meaning it's the control, meaning it's hybridization within itself, to itself. No? So, of course, in that first hybridization experiment, we will have 100% level of radioactivity because it has the same, okay, it hybridizes with its own DNA, right? It's the control, so it's 100% level of radioactivity and level of hybridization. So that's for your control. That means because it's the DNA of organism 1 hybridized to the DNA of organism 1, of course, it will surely have a 100%, and that would mean it's the same strain of same species. Kasi nga, this is the control. Now, for the second hybridization experiment, we're going to use 1 times 2. We're going to hybridize the DNA of organism 1 with the DNA of organism 2. And here, we will look at the percent level of radioactivity and the percent level of hybridization. Now, if it has, for example, it turns out that there are only 25% level of radioactivity or hybridization, now we can see the amount of the hybridization, no, between these 2 different organisms. So what can we conclude about that? What are the threshold values that we're looking for? What can we say about this? Why are we saying this is the same species? Why are we saying this is a different species? Okay, so what are the threshold values? So let's just go here. So the threshold values for classification, of course, for the 1 times 1, if it has 100%, it's always the same species again. From 70 above to 100, if the percent DNA hybridization is 70% to 100, you can conclude that it is from the same species. Now, if the percent identity, if the percent DNA hybridization is only about in between, 70 and 25 degree at 25, you can only say that they are of the same genus. Okay? They are of the same genus. But they are different species. They came from different species. Based on that percent hybridization. Based on that, again, this is based on the level of radioactivity, right? Because we're using phosphate, radioactive phosphate as labels here. And if the percent hybridization in the 1 times 2 is less than 25%, you will say that they are from different genera and they are from different species too as well. Okay, so that's how you use and interpret the hybridization experiment that we're doing here. Okay, so this is how you interpret the result for a DNA hybridization experiment. It's powerful because you're using the whole genome. You hybridize the whole genome of your organism. Okay, we only look at the 1 times 2 organism, the hybridization experiment for organism 1 and organism 2. And we conclude from that percent hybridization. Okay, so that's genomic hybridization. Next is the GC ratios. So as you have remember from the Chargaff rules, A to T, G to C, there's always this certain base pairing that is always being followed. Now, we use that, and more specifically, we use the ratio of your guanine and cytosine in the nucleotide bases of the DNA sequence, and use that as a basis, a very rough estimate of the relative relatedness of this species that we're looking at. So, how do we conclude? It's not here. How do we compute for the GC ratios? For example, how do we compute for the GC ratio? So, GC ratio is equal to, of course, we have your Chargaff rule, so it's easier to compute for this. So, we have G plus C over the total amount of bases. The total amount of bases. These are the G, of course, plus C, plus the T, plus the A's. Right? So you have remembered times 100%. No, times 100. Yeah. So this is how you compute for the GC ratios. So, for example, I gave you the organism has about 100 G guanines. How many cytosines are there? Of course, there's also 100 cytosine. So that's 200. I gave you at least 250 adenines. How many thymines are there? It's also 250. Because G to C, T and A. Okay, don't forget this. GCTA. Okay? And how do we interpret the GC ratios? We just look at this. If the GC ratio is more than 5%, okay, they have few. We can confirm that they have few DNA sequences in common and therefore are unlikely to be closely related. Right? They are unlikely to be closely related. So if you have greater than 5%, if you have greater than 5% difference in the GC ratio, you can conclude that they do not belong to the same species. So that's how you interpret GC ratios, the results in GC ratios. But as you can see here, there is a big problem with using GC ratios, right? You can see it clearly right away because it doesn't always mean that they have the same amount of GC. They have the same amount of GC. They have the same GC ratio. It doesn't always mean that they are the same species. Because, for example, if you have organism A and organism B that has both GC ratio of 46% and 46%, they may still, there's still this chance that they may belong to different species and they are not related at all. Because GC ratios just measures the amount of the G's and the C's, the G and C's in the DNA. It doesn't give you the specific sequence, right, of that G and C's in that DNA. Do you get that? That's why I always tell my student that the GC ratio are just an estimate or just a rough index of the relativeness, the relatedness of your different microorganism. The last one for genomic analysis is what we call the use of DNA profiling methods. In DNA profiling methods, we generate what we call DNA fragment patterns that we can use for genotypic analysis and comparing similarity among different species. Okay? So one example of DNA profiling method is what we call ribotyping, wherein we create what we call the ribotype. So what are ribotypes? Ribotypes are we call the DNA fragment patterns. We call the DNA fragment patterns the image of the DNA fragment patterns, the ribotype. Okay? So the ribotypes are the DNA fragment patterns that we create. Yun pala siya. The DNA fragment patterns. But just like your karyotype, right? In your karyotyping in your Bio 140, karyotype are like this, the image, fingerprint of your chromosome. In ribotype, it's the image fingerprint of the different DNA fragment patterns of your microorganism. Okay? So how do we create a ribotype? How do we do ribotyping? The first is we need to extract the DNA. And in that DNA, we're going to cut it into smaller pieces using what we call restriction enzymes. Best example of a restriction enzyme is your ECO R1. So ECO R1, if you have a DNA of your microbes right here, it's a long DNA, just imagine this is a long DNA. If you have ECO R1, you put it in the mixture, what will ECO R1 do is it will cut your genome into certain location in what we call restriction sites. Okay? it will create a cleave. Echo R1, for example, will create a cleave on certain restriction site of your organism, of the genome of your organism. Thus, it will create many fragments of your DNA. And we call that fragments your DNA restriction fragments. Okay? And the specific genes that we're using here, that we're cleaving here, is usually on the 16S and the 23S rRNA DNA, RNA gene of your organism. Okay, so let's go at it again. So, we have 3 different strains of bacteria here. What we'll do is we'll extract the DNA. After extracting the DNA, we'll use a restriction enzyme. For example, the restriction enzyme here that we're using is Echo R1. So, a brief background on Echo R1. Echo R1, the E in Echo R1 is Escherichia from the E. coli, right? Escherichia. Okay? The co is coli because E. coli. The R in the Echo R1 is the strain, so here the specific strain is RY13, and the I in Echo R1 is what we call the number, okay? This is because Echo R1 is the first restriction enzyme isolated from the strain. The first enzyme, no? The order of the enzyme that was isolated from this RY13 strain. Okay, so that's Echo R1. Again, Echo R1 is a restriction enzyme, an example of a restriction enzyme. What's good about Echo R1 is that when it encounters the restriction site, this specific restriction site right here, it will introduce a staggered cleave. It will cleave that DNA. So if Echo R1 encounters GAA-TTC and the complement CTT-AAAG, what it will do is it will introduce a cleave right here and there. It will introduce a staggered cleave on a staggered cutting pattern on the DNA. So that's how Echo R1 introduces a cut in your DNA. Once our restriction enzyme encounters this specific restriction site on your DNA, it will introduce a staggered or a zigzag cleave, a staggered cutting pattern on your DNA, where there will be what we call sticky ends on this portion, or what we call overhangs. Okay? Sticky ends or overhangs. Another thing to mention with our restriction site is that it is palindromic in nature, meaning it reads the same as backwards and forward. Okay? It reads the same as backwards and forwards, your restriction site. After application of your restriction size, of course, you will have different lengths of fragments of your DNA. And of course, that differing length of your DNA or your DNA fragments, DNA restriction fragments, would have different travel rate when we do your electrophoresis. When we subject it to electrophoresis, the smaller DNA, okay, the smaller DNA fragments would travel faster than your larger DNA fragments right here. So it will have this different, the DNA fragments will be separated by their size. After being separated using gel electrophoresis by size, we will use a nylon filter and we'll transfer that DNA into that nylon filter. We're now going to use what we call DNA probes to hybridize with those DNA fragments. So what are DNA probes? DNA probes are short single-stranded DNA that is complementary to a particular sequence of your DNA. So there are two types of DNA probes that we're using here actually. We have your 16S rRNA and your 23S rRNA gene probes. So these are the two gene probes that we're using. So what's going to happen when we use those gene probes is that your gene probes will only adhere to certain regions of your DNA fragments that have a complementary sequence. Thus, it will only create binding patterns on certain regions. And thus, it will create what we call the ribotype profile. So this picture right here is our ribotype. Okay, this is the ribotype now. So, what can we see in here? So, always in your ribotype, no? More often than not, the number one well right here, we put here in this portion what we call the DNA ladder. Okay? So we put in number one, the first well of your ribotype, the DNA ladder. What do we mean by a DNA ladder? This DNA ladder is, well, a DNA that has specific bonding patterns that reflects different sizes of DNA. So your DNA ladder will tell you that the first bond is a certain size of KiloDalton or so on and so forth. So that's your DNA ladder, the first well usually. So your DNA ladder, each band in your DNA ladder means a specific molecular size in KiloDalton usually. Okay? In KD. Now, of course, we're just going to use the DNA ladder as a comparison of the molecular size of the DNA fragment. Again, the closer it is to this side, the well, okay? You have wells right here, remember? You have wells right here. There are wells right here where you put the DNA, okay? So you have wells right here. The closer the DNA fragment is to the well, of course, the larger the DNA is. The larger the molecular size. The farther it is, the smaller the molecular size of the DNA. Okay? Now, what we're going to look at your ribotype is the difference in bonding patterns across your organism. So, how do we conclude the degree of similarity among organisms? We can say that these two, for example, are the same species if they have the same bonding pattern. Okay? But these two species, on the other hand, are not the same because this organism 7 lacks the bands here and bands right here. And also, it's not perfectly aligned together. Okay, so that's how you interpret your ribotype. The cool thing about ribotype versus DNA, whole genome DNA hybridization is that your ribotype profile uses only your 16S and 23S rRNA gene probes. So it is more easily concludable. No? You can conclude more easily using ribotype because you're using not the whole genome but only a portion of the genome. You're using only the 16S and the 23S. So it's more easily to conclude on, and your conclusion thus will be more accurate using the ribotype than using a whole genome to identify the organism, to compare the organism. Okay? So that's how you use ribotyping. We have now come to the last portion of your lecture in Polybasic Taxonomy, wherein we do phylogenetic analysis. The first step in phylogenetic analysis is to obtain the DNA, extraction of DNA. After extracting the DNA, we'll do sequence alignment using BLAST. It's a web-based algorithm, basic local alignment search tool. You can visit it using this link right here, where what it does essentially is it aligns sequences automatically. Thus, it can identify genes and compare different sequences from their database based on percent homology or the number of hits that your sequence that you provided to their database, to their vast database that they have. After BLAST sequence alignment, you will do we will do now a construction of your phylogenetic tree. Here we use multiple numbers of sequences and create a graphical description of the relationship and the position of your organism on the phylogenetic tree that you're making. So basically, the flow through for your phylogenetic analysis contains these different steps, the 3 different steps. The first one is the isolation of DNA. Okay, after isolation of DNA, you will only use certain portion of your DNA, not the entire portion. For example, we will use, for bacteria, specifically, we use the 16S gene. For fungi, for example, we use the ITS, internal transcribed spacer, okay, for fungi, and so on and so forth. Different organism uses, groups of organism uses different genetic marker, but that depends on your organism of choice. So for bacteria, we uses we only amplify the 16S gene. So we will only amplify that certain portion using what we call the PCR, polymerase chain reaction, where we will have your forward, reverse base, the forward and reverse primers, and then essentially PCR is a good technology that amplifies your DNA. So it will amplify your gene, this DNA portion. It will create exponential copies of multiple copies of that of that gene, and then we'll check the size of that. If we have captured or amplified the right gene using your agarose electrophoresis, gel electrophoresis, we will check the KiloBase or the molecular size of your amplicons. So these are your amplicons, because we have amplified it from the PCR reaction earlier. So we have your amplicons. What we'll do now with the amplicon is we'll subject it to sequencing. This is the second step. So what we do in sequencing, there are a lot of sequencing technologies out there. We have your Illumina sequencing, we have Pyro sequencing, we have Sanger sequencing. All of those sequencing processes, you will encounter it in your molecular biology, but there are a lot of sequencing facilities, sequencing technologies out there. But what it does, it identifies the specific sequence, that's why sequencing, of your gene, of your DNA. So after subjecting your amplicons, your PCR amplicons of your specific gene that you're using, okay, it will look something like this, where you have what we call a chromatogram of the different peaks that correspond to certain bases. So we'll now have your text file or FASTA file of your sequence, okay, of the sequence. So usually the chromatogram file is in ABI file. You'll convert it to a FASTA file, and this FASTA file format, you can then input it in your BLAST, the web BLAST program, a sequence alignment program in the web, in NCBI database, and compare your sequence to the database of sequences of different organisms across the world. So if there is a high similarity of high hits in that BLAST sequence of your organism, more likely it will have a high % identity. And that % identity is the one that we're using in describing the identity of your organism that you have isolated. Now, after doing BLAST, you will gather all of your FASTA file and create what we call your tree. So this is the last portion. We'll create a tree, no, a phylogenetic. We'll construct a phylogenetic tree based on the FASTA sequences file that we have created. We can also download different sequences from different organisms across that NCBI database and also create a reference distinct species, no? a reference species, an outgroup, an outlier for our tree making process. Now, if you have been in my Bio 110 class, you may have encountered how we deal or how we produce the flow through of the phylogenetic analysis process, how we isolate DNA, how we create sequence alignment, how we generate the tree. But if you haven't yet taken your Bio 110, I suggest you visit my YouTube account and go to the YouTube account of Sir Patrick and find Activity 2, Basics of Mega Software. You can read on the description right here. It's a very simple description. You can also access the sequences right here if you want to practice on it. So these are the sequences that I am using. So this is a relatively long and long video because it's an activity for Bio 110 back then. But yep, you can go on the timestamp because you can go to certain timestamp. If you want to skip the intro portion, you can go ahead and go to the certain process that we're doing right away, how we complete the forms, how we identify what to do. Okay, so it has timestamp right here, the filling up of order forms, the trimming up the sequences tracer file, config assembly, blast search, how we do blast search, how we download the outgroup, muscle alignment, the tree making process is also here. So if you want to study more on that, you can use my the video that I created, the activity, and you can download the files, the raw sequences right here if you want to practice on that. But if you have already done that, that's good for you. You're good to go. Okay? So yeah, we also do blast here, right here. If you have forgotten how to do blast search, just just watch the video on how to do it. It will guide you clearly on how to do it. And we look on the percent identity and the e-value. So basically, we want a higher percent identity for us to conclude the the identity of the isolate that we're playing with, that we're dealing with. But we also check the accession number. We also check the source of that organism, because sometimes the sources of the organism differs. For example, sometimes the organism that you have isolated from, for example, in school, in the air in school, you have isolated your organism of your interest from the air here at school at UPB. But when you check the accession number here of the organism that the high percent identity, the organism that showed in blast search, during blast search, when you click the accession number, it says there it was isolated from geothermal vents. That's a big red flag. Okay, it's because your sources, the sources of your organism is from campus air, and the one that is mentioned here is from geothermal vents. So that's a big red flag for you. So take note of that too, for you to easily conclude or identify, so that you don't get confused on how to identify your organisms. Anyway, so I hope that video will just go to the specific timestamp that you want to view on, so that you don't get too flustered due to the length of the video. But yeah, you can practice also using this sequence right here, our Mucor sequence. You can copy this sequence and perform BLAST search. Just search BLAST, input BLAST in Google, click Basic Local Alignment Search Tool of NCBI, click Nucleotide BLAST, okay? And in that portion, input your sequence right there, right here. After inputting your sequence right there, I think the interface didn't change that much. After inputting the sequence, you need to change nucleotide correction right there. Okay. Okay. You can also, it's up to you if you want to take sequences from type material. No? Here nucleotide collection. I click the nucleotide collections right here too. And so on and so forth. You can explore it more often. And then you click BLAST. After clicking BLAST, you can also tick here, highly similar sequences. This is what it will show you. Something like this, but it's more, it's improved now. It has a different look to it, the interface. You can click the accession number and look at the percent identity right there. And you can also view my video again to create this phylogenetic tree right here. I created this tree back during, I think, 2006 when it was still Mega 7 software. Now we're using Mega 10 software na. Okay, so yeah. Again, the important thing to note here, back here, is the percent identity. You need to look at the percent identity. You need to have a high percent identity. You need to have at least 97 and above percent identity for you to conclude if that is the same organism as your sequence from the database. And for the phylogenetic tree construction naman, you need to look at the what we call bootstrap value. Again, how do we conclude the meaning of this bootstrap value? So essentially, when you have a 1000 bootstrap replication, okay, that, and you have, so during the settings of your tree, you basically told the software to do 1000 bootstrap replication. Now, when you look at this tree right here, and it has 21 bootstrap value in this portion, it means that in that 1000 bootstrap replication, 21% of the time, so 21% of 1000, I don't know what, 210, I guess. Anyway, so 21% of the time, these two sequences clustered together. So when we look at this naman, 80%, so 800 times during that 1000 bootstrap replication, these people clustered together naman, 80% of the time. 800 times, they have the software found that these three always clustered together. Okay? So that's how you you interpret it. Because remember, the sequence alignment and this tree making process is based on sequence homology. It's phylogenetically informative, meaning it gives info about the organism's divergence from a common ancestor just by looking at the mismatches in that DNA. That could be due to, of course, your mutation, your deletions, your insertions, and so on and so forth. Okay? So for bootstrap value naman, usually we accept at least 70% bootstrap value and above. But of course, the higher the better. Okay. So lastly, we need to mention this here. So again, for DNA hybridization, the species concept in microbiology, for a thing to be the same species, it needs to have at least 70% or greater DNA hybridization, 70% or higher. For The 16S RNA gene for the BLAST sequence na ginagawa natin, the earlier mentioned the BLAST search here, right here, the BLAST search right here, we need at least 97% or greater. We need to have at least less than 3% difference in the 16S RNA gene sequence for us to say that they belong to the same species. Okay? So that's how we define a species, a bacterial species here. Okay? It's a strain sharing a high degree of similarity, both in your DNA-DNA hybridization or your 16S RNA gene sequence. And if you want to dwell more on your what we call taxonomy of microbes, this is how we do it. How do we describe that species? We need to include phenotypic characters, the proposed name, the BLAST sequence, and then we must also deposit a viable culture into at least 2 international culture collections. Just like for plant that has herbarium or natural museum for your animals, I guess. For microbes, we can deposit it to certain microbial culture collections. For example, in Belgium, France, Germany, Japan, Netherlands, Scotland, and USA. No? So these are different microbial culture collections if you wanna deposit your collections right there. So that's it for today. Thank you for listening. And once again, this is Sir Patrick signing off.